We'll go over the processes of HIV attachment and entry into the host cell and integration into the host genome. To start, we have our HIV particle with its outer membrane, capsid, and the red squiggly line is its RNA genome, and it has its fusion peptide and GP120 exposed. We'll use these peptides to attach at the host cell membrane, with GP120 attaching to CD4, and it will need the CCR5 coreceptor unless it is a late infection, in which case it will use CXCR4 instead. Once these attach, the fusion peptide is allowed to do its very self-explanatory job of fusing the host and viral membrane. The fusion peptide will pull the membranes incredibly close together, causing them to fuse, and the HIV membrane will merge with the host cell membrane, spitting out the capsid and genome into the host cytosol. Once inside, the capsid dissolves and we are left with only the squiggly line that is the HIV genome. Upon closer inspection, the red squiggly line is revealed to be composed of several parts. The R, U5, PBS, the open reading frame, PPT, U3, and R. For this part about converting it to double-stranded DNA, RNA will be represented with red because they both start with R, and DNA will be represented with blue because it's not red. Anyways, to begin this process, tRNA will bind at PBS, and this will allow translation, reverse translation I should say, of U5 and R with RT and RNase H. So it'll give us R' and U5', but will cause us to lose the R and the U5 portions of the original genome. With the loss of this RNA, the newly synthesized DNA falls off, but fortunately there is another R for it to bind to with R'. So it does, and this readies more reverse translation. So reverse transcriptase and RNase H come through and do their thing, and give us U3', PPT', ORF', and PBS', and they degrade the original strand except for PPT, because PPT resists degradation by RNase H, which will be important for the next step. The PPT RNA is then used as a primer for synthesis of DNA complementary to U3', R', U5', and PBS'. But since PPT is only resistant to RNase H, not completely immune to its effects, it gets chewed up and we lose the original PBS primer as well. This causes the new strand to detach and reattach over at PBS'. Now the cell has the ability to use both PBS and PBS' as primers for more synthesis. So on the bottom strand we get U3', R', and U5' synthesized, and on the top strand ORF, PPT, U3, R, and U5 all synthesized. Now we have a complete molecule of double-stranded DNA, complete with long terminal repeat signature of retroviruses. In this case it would be the U3, the R, U5, and then U3, R, U5 on the other side as well. If we zoom back out, we will see that our original red squiggly line has been replaced now by two blue squiggly lines representing the double-stranded DNA. This double-stranded DNA is then trafficked through the host cytosol into the host nucleus, and then integrase will take this DNA, cleave a spot in the host genome, and insert the viral double-stranded DNA into the host genome.